The overall goal of this procedure is to generate and characterize T lymphocytes from induced pluripotent stem or iPS cells. The iPS cells can then be differentiated in vitro on an OP9DL1 culture system and then matured in vivo in a RAG-deficient mouse. Or the cells can be genetically engineered to overexpress OT1 TCR and then adoptively transferred for in vivo development. After 6 weeks of in vivo differentiation, the mice can be challenged by intraperitoneal injection with EG7 tumor cells, and then the antigen specificity of the iPS-derived T cells can be evaluated. Ultimately, the iPS cells can differentiate into both conventional and antigen-specific T cells, the latter of which are able to protect animals from tumor challenge.
The implications of this technique extend toward individualized cancer immunotherapy. The method allows the generation of large numbers of tumor-reactive T cells derived from minimal amounts of patient's blood or skin tissue.
On day 0, transfer 5 × 10⁴ iPS cells onto a 100 mm culture dish containing a confluent OP9DL1 cell monolayer in 20% FBS alpha MEM media. On day 5, trypsinize then centrifuge the cells. After incubating them on a fresh 100 mm culture dish for 30 minutes in 37-degree incubator, plate 5 × 10⁵ of the floating cells in 20% FBS alpha MEM media and mouse flat3 ligand on a new 100 mm dish containing a monolayer of OP9DL1 cells. On day 8, gently pipette down the loosely attached cells, centrifuge the cells, and then transfer the cells to a single well of a 6-well culture plate coated with confluent OP9DL1 cells. Incubate the cells at 37 degrees Celsius, 5% carbon dioxide for 2 more weeks. On day 22, incubate detached iPS cells in fresh media on a new culture dish at 37 degrees Celsius for 30 minutes. Then remove about half of the floating cells and pass them through a 70-micron nylon strainer to exclude cell clumps and wash the resulting single-cell suspension 3 times with cold PBS. After the third wash, resuspend the cells in PBS at 1.5 × 10⁷ cells per milliliter and maintain the cells on ice. Before IV injection, place a 4-week-old RAG-deficient mouse under an infrared light to dilate the tail vein. Then fill a 1 milliliter syringe equipped with a 26.5 gauge needle with 200 microliters of the cell suspension and adoptively transfer the cells through the dilated tail vein. 3 weeks later, after confirming euthanization of the adoptively transferred mouse, remove the lymph nodes and spleen. After mechanically breaking down the tissues, lyse the single-cell suspension with ACK lysis buffer, and then wash the resulting mononucleosides twice in cold PBS. Then, after staining for cell surface markers, evaluate the cells by flow cytometric analysis. On day 0, use the gene gamma transfection reagent to transfect overnight plated Plat E packaging cells with an OT1 MIDR. On day 1, seed 1 × 10⁶ iPS cells into 1 well of a 0.1% gelatin pre-coated 24-well plate. On day 2, collect the pseudovirus-containing supernatant from the Plat E cells and then pass it through a 0.4 micron filter to exclude potential contaminants. After centrifuge transducing the cells in the presence of polybrine for 1 hour at 330 × G at 32 degrees Celsius, incubate them overnight at the same temperature. After repeating the transduction procedure, trypsinize the transduced iPS cells on day 4, and then, after pelleting the cells, resuspend the cells in fresh media and seed them on pre-coated irradiated SNL767 feeder cells. Once the transduced cells have reached confluency, sort the GFP and DS-Red double positive cells by flow cytometry. 6 weeks after the adoptive transfer of the iPS cells, inject 50 microliters of EG7 thioma cells into the peritoneal cavity of the same mouse. On day 50 of the tumor challenge, use a Miltenyi Biotech CD8-positive T-cell isolation kit to sort CD8-positive T-cells from the spleen and lymph nodes of the adoptively transferred animal. Mix the isolated CD8-positive T-cells with irradiated splenocytes from a naive C57 Black 6J mouse at a 1:10 ratio and pulse the co-culture with 0.5 micromoles per mil of ova for 40 hours. Thereafter, add Berfeldin A to the cells for another 4 hours. Finally, evaluate the cell populations by flow cytometric analysis. After isolating splenocytes from a naive C57 Black 6J mouse, split the cell suspension into 2 groups. Label the CFSE high group with 5 micromoles per mil of CFSE and pulse the cells with 10 micrograms per mil of ovapeptide for 1 hour. Label the CFSE low group with 0.5 micromoles per mil CFSE and do not pulse. Mix 2.5 times 10 to the 6th CFSE high cells with 2.5 times 10 to the 6th CFSE low cells in PBS. Then analyze the splenocytes by flow cytometry for CFSE expression 16 hours after adoptive transfer into the mouse of interest. To count intraperitoneal tumor cells, on day 20 of the tumor challenge, use a 10 ml syringe equipped with an 18.5 gauge needle and cold PBS to lavage the peritoneal cavity. Count the recovered tumor cells. For identifying tumor-infiltrating cells, remove the tumor at a late stage of the tumor challenge and then cut the tumor into 3 pieces. Place the first tumor piece into a cryovial and place the vial on dry ice immediately. Fix the second piece in formaldehyde. Preserve the third piece in conditioned RPMI 1640 media. After air drying the cryopreserved tissue sections, fix them in cold acetone for 15 minutes. After air drying the fixed sections for another 15 minutes, wash the slides for 5 minutes in PBS. After the wash, place the slides in a moist chamber and cover the tissue sections with 30 microliters of 3% BSA in PBS for 30 minutes to block nonspecific binding. Then, blot off the blocking buffer and incubate the tissue sections with a 50 microliter mixture of PE anti-TCR V-alpha-2 antibody and FITC anti-OVA antibody diluted in 3% BSA in PBS in the moist chamber for 2 hours. At the end of incubation, wash the slides 3 times in cold PBS. Finally, mount the sections with a water-based mounting media before fluorescent microscopic examination. For flow cytometric analysis of tumor-infiltrating T cells, squash the tumor into a single-cell suspension. Then, analyze the cells for specific surface markers. CD3 and TCR-beta are used as markers of T cells. To determine whether stimulation of iPS cells with the Notch ligand DL1 could contribute to T-cell differentiation, CD4 and CD8 cell surface expression on CD3-positive TCR-beta-positive iPS cell-derived cells was assessed. Note the dot plot on the right, which shows day 22 CD3-positive TCR-beta-positive, CD4-negative, CD8-positive, single-positive T cells generated from iPS cells in vitro. In addition, the iPS cell-derived single-positive cells from the previous figure produced interleukin-2 and interferon gamma when stimulated in vitro by plate-coated anti-CD3 and soluble anti-CD28 antibodies, confirming that the iPS cell-derived T cells are functional. After adoptive transfer into recipient mice, the majority of TCR gene-transduced iPS cells underwent differentiation into CD8-positive CTLs, which responded in vitro to peptide stimulation by secreting interleukin-2 and interferon gamma. In these dot plots from pooled lymph node and spleen cells, CD8-positive V-beta-5-positive T cells were generated in mice adoptively transferred with TCR-transduced but not control-transduced iPS cells. The CD8 positive V-beta 5 positive populations were positive for intracellular cytokine staining for interleukin 2 and interferon gamma production, represented in these histograms by the dark lines, as compared to the shaded isotype control histograms indicating that the iPS-derived CTLs were functional. These data show that CFSE high cells pulsed with ovapeptide, represented by the peaks on the right in each graph, and CFSE low control cells, represented by the peaks on the left, that were injected into mice 10 weeks after iPS cell transfer or 1 day after OT1 CTL transfer. The antigen-specific iPS-derived CTLs responded to antigen stimulation and had cytotoxic activity as indicated by the absence of CFSE high cells. Here, OT1 TCR gene-transduced iPS cells were adoptively transferred into C57 Black 6 mice, and then the mice were subjected to challenge with EG7 tumor cells. On day 20, tumor cells in the peritoneal cavity were enumerated. Note the significant reduction in tumor cell numbers in mice that received TCR-transduced iPS compared to the control groups. Most importantly, adoptive transfer of TCR-transduced iPS cells triggered infiltration of ovoreactive CTLs into tumor tissues and protected animals from tumor challenge. As seen in this H&E staining of tumor tissues recovered days 30 to 35 after tumor challenge, tumors from mice adoptively transferred with TCR-transduced cells, or OT1 CTLs, but not mock-injected or control transduced cells, were infiltrated by inflammatory cells as indicated by the red arrows. Here, ova-specific V-alpha-2 positive CTLs, in red, were found to infiltrate ova-expressing tumor tissues, in green, while tumors in mice from the control groups had fewer or no tumor-infiltrating cells. In this figure, single-cell suspensions from tumor tissues were analyzed for expression of V-alpha-2 positivity and V-beta 5 positivity by flow cytometry after gating on the CD8 positive population. Note that tumor tissues from mice adoptively transferred with TCR-transduced iPS cells exhibited the highest level of tumor-infiltrating cells, while tumors from mice that received control-transduced iPS exhibited no infiltration by ova-specific CD8 positive T cells.
After watching this video, you should have a good understanding of how to generate antigen-specific T cells from iPS cells and how to evaluate the functions of the iPS-derived T cells.